Hi everyone, good evening guys. Welcome back to Unacademy NEET English channel. My name is Tina and I'm your biology educator. And guys, in today's session, we are going to complete Human Physiology unit-wise final revision for NEET 2026. We are going to complete our Human Physiology. Quickly get ready with your copies and your NCERTs to make quick notes and quickly share the session with all your friends. Yes, good evening all of you. How are you all? How's your preparation going on? Please let me know in the chat. I am very curious to know that. And I hope guys you know that NTA is releasing questions in the NTA WhatsApp group. So make sure that you all join that. And also from Friday, okay, let me give you a very important information. From this Sunday onwards till next Friday, that is 26th of April to 1st May, guys, we are starting AIMT. We are going to celebrate this AIMT week where you will be solving the mock tests every day from 2 to 5 p.m. So let's get NEET ready before the exam and solve all these mock tests. You'll be getting all the video solutions for them. So NTA is telling you indirectly by giving questions that you have to practice questions. I hope everybody's ready. Give me a thumbs up so that we can start with the session quickly. I will be taking a marathon session where I'll be completing more chapters. Rajat, I'll be doing that. Good evening, guys. Good evening. They're telling notes will be uploaded in the but they are not telling which group. Which group? It is Unacademy NEET English group. What else? It's easy. You can easily find it. Just write down Unacademy NEET English. You will be able to find out this group. This is the Telegram group, and this is where I upload all the hello, good evening. All right, guys, let's get started. We have a lot of chapters to complete today. I am going to start with the first chapter of Human Physiology, that is breathing and exchange of gases. So starting with, okay, let's get started, guys. We have a lot of things to do today. Lot of things. Start with breathing because I'm coming after 30 minutes. Please, coordination last. Yes, Ansh, I am starting with breathing and exchange of gases only. Now, let's get started, guys. Talking about the respiratory organs in rest of the organisms, see, we have already discussed animal kingdom. Let's have a quick revision. In sponges, coelentrates, flatworms, it occurs through diffusion, through the body surface. They don't have any specific structure. For earthworm, it is the moist cuticle or the skin, and we call such respiration as cutaneous. We call it as cutaneous. For insects, it is the tracheal system. For aquatic arthropods and mollusks, we have gills. Through gills, respiration is known as branchial respiration. For terrestrial organisms, we have lungs, which is called pulmonary respiration. And if we talk about amphibians, guys, for example, frog, then it shows respiration through lungs, skin, as well as mouth. This is so it uses skin while it is in water, while lungs and mouth are used while it is on land. While the tadpole stage of frog is going to respire through gills, it is fish-like. I hope these points are clear. Now moving on to just a second. Yes, the human respiratory system. So human respiratory system, as we know, starts with the nostrils, the nasal passage, the nasal chamber, moving into the pharynx, which is a common passage, then glottis, which is the opening of larynx, then trachea, and then from trachea it gets into the bronchi. And from bronchi it goes into the bronchioles, and from bronchioles ultimately it's going to move into the alveoli. This is the unit of lungs where actually gaseous exchange is going to occur. Right now, this whole air passage is divided into two parts, which is called conducting part and exchange part. Now we know larynx is the voice box. It is the voice box, and it is basically a cartilaginous box made up of 9 cartilages. Now talking about your trachea, it is the windpipe, and it is a straight tube up on the which side it is. It is on the dorsal side of your esophagus in the mid-thorax region, and right at the 5th thoracic vertebra it gets divided, guys. It gets divided. So this is the limit where it gets divided, and the location is fifth thoracic vertebra. Fifth thoracic vertebra. You can write it T5. Is this point clear? Now, it gets divided, and now trachea, see, it is trachea, then, just a second. Yes, trachea, then bronchi, and then bronchioles. They are all covered by C-shaped cartilaginous rings. This cartilage is haline in nature. Is that clear? The cartilage which covers this is incomplete C-shaped cartilaginous rings. Why is it present? To prevent the collapse of these ducts. Are these points clear to all? Are these points clear to all? Ma'am, biology notes is not in that biology notes. I'll check, Mahima, once again. I'll check. Okay. Now, moving on to the alveoli, moving on to the unit of lungs which is known as alveoli. Guys, please focus here. Unit of lungs which is called alveoli. Now, alveoli are the thin, irregular vascular sacs, highly elastic, large surface area, rich in blood vessels, rich blood supply. So all these things help it in being the unit of lungs, being the perfect position where gaseous exchange can take place. Is this clear? Now, as I said, the whole respiratory tract is divided into two components. One is the conducting part, and the second is the exchange part. Now, see, the conducting part, this is the exchange part. Conducting part and the exchange part. So conducting part is from external nostrils till terminal bronchioles. So from external nostrils till terminal bronchioles is your conducting part, because here only the air is conducted. No exchange of gases is taking place. While alveoli along with the alveolar ducts, these two structures, they constitute your exchange part, where actually gaseous exchange will take place. Is this point clear? So external nostrils till terminal bronchioles conducting, and alveoli and their ducts form the exchange part. Now see, what are the functions of the conducting? Other than air conduction, it also purifies the air, it removes the dust particles, it adds moisture to the air, humidifies, and makes sure that the air is at the body temperature. I hope these points are clear. Now, guys, lungs are located in a specific thoracic chamber. It is an airtight thoracic chamber. Lungs are also protected by the covering. This is a protective covering of lungs. Protective coverings of lungs are known as pleura. As said, it is double in nature. The outer one is known as parietal and the inner one is known as visceral. And in between the two is present the pleural fluid, which reduces friction. Are these points clear to everyone? Please let me know. Now, this thoracic chamber, that means lungs, they are covered, they are surrounded by different structures. So what is the thoracic boundary? So lungs are surrounded by different structures. See, on the dorsal side, back side, we have the vertebral column. On the ventral side, we have sternum. On the lateral side, we have ribs. And on the inferior side, on the lower side, lies the diaphragm. Now, let's come on to the function. Of lungs, which is gaseous exchange, right? So for gaseous exchange to take place, it has to breathe. So breathing is that first step of where gaseous exchange occurs between atmosphere and lungs. So this is a gaseous exchange between atmosphere and lungs. It is also known as ventilation or pulmonary ventilation. Are these points clear? Now see, gaseous exchange is always because of the pressure difference. So air moves from high pressure to low pressure area. Air moves from high pressure area to low pressure area. Now see, if we want air to enter into the lungs, so within the lungs there has to be negative pressure. Remember that. So we can't manipulate atmospheric pressure, but we can manipulate the pressure inside the lungs. Now see, if we want to change the pressure, we can't do that directly. We have to change the volume. Are you getting that? So air will move towards the lower pressure area. You want to inhale, so lungs should have negative pressure, right? So intrapulmonary pressure should be low. Now in order to decrease the pressure, we have to increase the volume, because pressure and volume are inversely related. I hope these points are clear. Now how are we going to increase the volume? How are we going to increase the volume? See, diaphragm will move downwards and it will become flattened. It will become flat. While ribs and sternum, they are going to be raised. So ribs and sternum, they are going to increase the volume in the dorsoventral axis, while diaphragm is going to move downwards and increase the volume in the anterior posterior axis. I hope these points are clear, right? So this is your anterior end, this is your posterior end. So when diaphragm is moving, so it is increasing the volume in the anterior posterior axis. Are these points clear? So overall volume of the thoracic cavity will increase and the pressure will decrease, so air will automatically move inwards. Now if we want the reverse to happen, that is in case of exhalation, so ribs and sternum, they move back to the original position, right? So the volume decreases in the dorsoventral axis. Diaphragm also moves backward and becomes dome shaped. Diaphragm becomes dome shaped, and this also reduces the volume. Now volume reduces means pressure increases. So volume reduces, pressure increases. Now the lungs have more pressure as compared to the atmosphere, so therefore air will move out. I hope all these points are clear. I hope all these points are clear to you. Please let me know. Right. Now, when we are talking about the forced breathing, guys, in case of forced breathing, other than, see, what are the structures which were used in normal breathing? What were the structures which were used during normal breathing? It was ribs. It was intercostal muscles. Intercostal muscles are the muscles between the ribs. And then we have diaphragm. And then we have diaphragm. But when we talk about the forced breathing, apart from ribs and intercostal muscles and diaphragm, there are two other structures which are used, which is, see, these are external intercostal muscles, but now we are going to use internal intercostal muscles plus abdominal muscles for forceful breathing. Is this clear? Now there is an instrument which is known as spirometer, or we also call it pulmometer, which helps in measuring the lung volumes. Let's talk about the lung volumes and the capacities. Pulmonary volumes and capacities, very important topic. Very important topic. Now see, first of all, when we talk about the volume, it is always a single entity. It is always a single value, while capacities are always more than two. It's always two or more than two. Multiple values will come together to form capacities. Now see, tidal volume. You have to note down the key words. See, tidal volume is during the normal breathing, normal inhalation, normal exhalation. So when you're normally breathing, it is a tidal volume. Inhale, exhale. Normally, you take in 500 ml, you throw out 500 ml. But if you have breathed normally, now exhalation is normal, but after normal exhalation, when inhalation becomes forceful, maximum, then that extra volume that you take in is known as IRV, which is 2500 to 3000 ml. That means 500 inhale, and extra that you can inhale, that would be 25 to 3000 ml. While expiratory is inhalation normal, but exhalation is forceful now. Now, in that case, the extra air that you can throw out other than 500, that is other than TV, that would be 1000 to 1100 ml. Are these points clear to everyone? Please let me know. And then we have residual volume. No matter how much forcefully you breathe, some amount of air, volume of air, that will always inside lungs, that always remains within lungs, is known as residual volume. It's also known as residual volume. Now coming on to the capacities. So we have inspiratory capacity. Inspiratory capacity is TV plus IRV. You must remember these formulae and just put on the values and find out the answer. Then expiratory is the total that you can exhale, that is TV plus ERV. Then functional residual volume, capacity. This is ERV plus RV. See, residual volume is when the volume of air remains inside lungs even after forceful exhalation. This volume will never come out after forceful exhalation, right? But functional residual capacity is volume of air that remains in lungs, after normal breathing. See, we are not always forcefully exhaling, right? After normal exhalation, the volume which will remain inside lungs, that is known as FRC. Then coming on to vital capacity. So vital capacity is IRV plus TV plus ERV. So both ways it is normal. Sorry, both ways it is maximum. So forceful exhalation followed by forceful inhalation. So when both the sides are maximum, both ways we are forcefully breathing, that becomes your vital capacity. And then total lung capacity is the total volume of air. Is that point clear to everyone? Please let me know. Please let me know. Yes, guys. I hope the tidal volumes are clear. Shall we move on now? Moving on to now the exchange. See, remember, exchange is going to occur at different places. So first of all, exchange occurred between atmosphere and lungs. When we say lungs, we are talking about alveoli, right? Next step would be exchange between alveoli and blood. And finally, there would be exchange between blood and tissues. Is this clear? So wherever the gaseous exchange is occurring, the basis remains the same. That is the partial pressure gradient. Gradient means difference has to take place. Is this clear? Now, you have to remember the pressure values at different places. You have to remember the pressure values at different places. Are these points clear? Okay. Now, coming on to the diffusion membrane. So when diffusion is occurring, so what are the membranes which are participating? See this diagram. So first of all, guys, the air is going to get in touch with the alveolar membrane, then go into the basement membrane, and then crossing the endothelium of capillary, it is going to pass into the blood, or the vice versa, right? From blood, it will cross the endothelium, move into the basement, and then again cross the alveolar wall, right? Now, each wall has to be only single layered for the diffusion to take place. Now, these three layers, alveolar epithelium, capillary endothelium, and basement membrane, together, these three layers constitute your diffusion membrane, and the overall thickness is less than 1 millimeter. Now, coming on to the transport of gases. See, while the exchange has been taking place, when it comes to blood, when the gases enter the blood is going to transport those gases, either oxygen or CO2. So, first of all, talking about oxygen transport. So, through blood, maximum oxygen is transported through hemoglobin forming oxyhemoglobin. And only 3% of it is transferred by getting dissolved in plasma. Is that clear? Now, hemoglobin, if we talk about, see, this is oxygen plus hemoglobin forming oxyhemoglobin. Now, one hemoglobin molecule can carry maximum of 4 oxygen molecules only. Is that clear? Is that clear? Now, the pattern in which oxygen and hemoglobin interact with each other, this can be studied through a special curve which is known as oxygen dissociation curve, ODC. Now, see the graph first. See, along the x-axis, we are taking partial pressure of oxygen in millimeter mercury, and along the y-axis, we are going to take hemoglobin saturation in percentage. Right? Now, if we plot the graph, first of all, the shape of the graph would be S shape, which is called sigmoid curve. Are these points getting clear? Are these points clear to all? Please let me know. Please let me know. So first of all, the shape of the curve is sigmoid curve. That is S-shaped graph. S-shaped graph. Why? Whenever we get a S-shaped graph for anything, see, there are always three phases. The first is always a slow phase. We call it a lag phase. Then comes the exponential or the log phase. And finally, we have a plateau or a constant phase. This is clear. Now, when we said that one hemoglobin molecule can only bind with 4 oxygen molecules, it can only bind with 4 oxygen molecules. In that case, guys, the first oxygen molecule will bind very slowly. So lag phase represents the binding of first oxygen molecule. Once the first oxygen molecule has bound to hemoglobin, the binding of second and third becomes very... All right, guys, all right. Connecting again. Okay, now this curve is not static. This curve can easily move to the right hand side or the left hand side. So this is the main line, this is the main curve, but this curve can easily move on the left hand side as well as the right hand side. So right hand side shift and left hand side shift means something. It has a significance. See, there are certain factors which are going to which are going to promote the right hand shift or the left hand shift. So first of all, let's understand what does RHS, that is right hand shift, indicate. So right hand shift indicates the the dissociation of oxygen and hemoglobin. This shows dissociation of oxygen and hemoglobin, while left hand shift shows oxygen and hemoglobin association, or you can say binding or binding. Are these points clear to all? So now what are the factors which will promote the oxygen hemoglobin dissociation? Whenever there is high pressure of partial pressure of CO2 or low, you can say low pressure of oxygen, low partial pressure of oxygen, or there would be low pH. Low pH means high acidity, high acidic conditions. Yes or no? Right? High acidic conditions are because of more CO2, then increased temperature. All these indicate decreased affinity of oxygen with hemoglobin, while left hand shift would occur in these conditions when the temperature is low, partial pressure of CO2 is low, but pressure of oxygen is high, and if oxygen is high, then pH would also be high. Are these points clear to all? So you must clearly remember these factors which are going to promote the right hand shift or the left hand shift. Are these points clear? Are these points clear to all? Please let me know. All right, guys. So we have already discussed sigmoid curve, hemoglobin affinity for oxygen, and so on. Now talking about the carbon dioxide transport. Now out of the total carbon dioxide, 70% is transported in the form of bicarbonate. So how is bicarbonate formed? So CO2 plus water will form what? Carbonic acid will form H2CO3. It will further dissociate to form H plus bicarbonate ions. Now these reactions are going to occur in RBC in the presence of enzyme carbonic anhydrase. So this is RBC. Now the same reaction is occurring in plasma also, but the difference is that carbonic anhydrase is the enzyme carbonic anhydrase is less. So same composition, same reaction is occurring here, but within the RBC, more bicarbonate ions are produced, but in plasma, less bicarbonate ions are produced. So since there is a difference set, these bicarbonate ions will move out in the plasma. Is it clear? And the place will be taken over by chloride ions. Now the bicarbonate ions which are moving out, they are going to help in transport in the form of sodium bicarbonate. They will combine with sodium ions and potassium ions and form potassium and sodium bicarbonate, and this is how the carbon dioxide transport occurs in the form of bicarbonate. Now where are they formed? They are formed in the plasma. They are formed in the plasma. Are these points clear? So, and then another 7% will be dissolved in the plasma. Another 7% of CO2 will be dissolved in the plasma. So in all, plasma helps in transferring in transport of 77% of CO2. Now out of 77%, 70% is in the form of bicarbonate ions and 7% is in the dissolved state. While rest of the 20 to 25% is going to transfer or get transported by hemoglobin forming carb amino hemoglobin. Please note down, with oxygen it will form oxyhemoglobin. With CO2 it forms carb amino hemoglobin, not carboxy. Carboxy hemoglobin is formed by carbon monoxide. Are these points clear to all? Are these points clear to all? So see, these are the reactions which are going to take place. Now the same reactions will be reversed at the tissue level. Are these points clear? Then coming on to the regulation of respiration. Regulation of respiration is of course done by your brain, but which part? It is medulla oblongata and pons varolii. So there's a special structure called respiratory rhythm center in medulla, and then another structure, pneumotaxic center in pons. Now it is clearly written in NCERT that more message or more impulse from pneumotaxic center is going to decrease the duration. It decreases the duration of inspiration. More the impulse coming from pneumotaxic center, lesser is the duration of inspiration. So please make sure that you know these lines. So pneumotaxic center is known as the switch off center. Actually, it is a switch off center for respiration. Are these points clear? Now, when is the rate of respiration going to increase or decrease? Now, this is going to be determined by two factors, two chemicals, which is carbon dioxide and bicarbonate ions. See, the fact is that it is the CO2 and H positive ions which regulate these two molecules. They regulate the breathing rate. So more the CO2 in your blood, more is the breathing rate. Less the CO2, less is the breathing rate. So it is not oxygen. Oxygen plays a minimum role in deciding, minimum role in determining the breathing rate. NCERT line, determining the breathing rate. I hope these points are clear. Now, the point is that how is CO2 and H positive ion detected? How is the level? How is the concentration detected? So we have some chemosensitive areas. Present in the body. So one is in the aortic arch, second is in the carotid artery. So these have aortic receptors. So these have CO2 and H positive receptors. Are these points clear? Are these points clear to all? Please let me know. So guys, we are almost done with breathing and respiration. Last not the least, let's talk about the respiratory diseases, starting with asthma. So asthma is basically the inflammation of bronchi and bronchioles, and the characteristic sound is that of wheezing. So bronchioles will be constricted. So of course, it is going to lead to difficulty in breathing. It will cause difficulty in breathing. And the characteristic feature is wheezing sound. It is allergic. It may be allergic response. Then coming on to emphysema. So emphysema, the main cause, see, it's a PYQ. The main cause of emphysema is always smoking. So what really happens? Smoking damages the alveolar walls, thus reducing the surface area. And it is a permanent change. So once the valve walls have been damaged, there is no coming back. Then next is occupational diseases. So guys, there are certain occupations which involve microfibers, which involve some microfibers. Now these microfibers, they keep on getting deposited in the lungs. For example, dust or cotton or asbestos, or it could be sand, right? Silica. So all the people who have like not the white collar jobs, I'm talking about the labor class actually. The people who are in direct contact with these microfibers, these microparticles, when they're continuously being exposed to it, and these molecules, these substances get deposited in their lungs, it results in fibrosis. So there is proliferation of fibroid tissue. Now this again decreases the surface area, leading to serious conditions. Now all these disorders which occur because of a specific occupation are known as occupational lung disorders. For example, silicosis because of silica, or asbestosis because of asbestos. Are these points clear? So guys, with this, the first chapter of human physiology is done. Please let me know, give me a thumbs up that you are done. How many of you have been uh, discussing this chapter with me? How many of you have joined the session since the beginning? Quick. Please let me know. How many of you have joined since the beginning and are done with the first chapter? Quickly. Aishu, Akila, Lakshya, Vaishwa, Mahima. All right. Perfect. Thank you, Aditya. Harini is from beginning. Okay. Cool, guys. Cool. Now, moving on to the second chapter. Second chapter. Perfect. Now, we are going to move on to the next chapter of human physiology, that is body fluids and circulation. Body fluids and its circulation. All right. Starting with this chapter. So, first thing first, which is the type of body fluids. So guys, we have overall two types of body fluids. One is blood, second is lymph. First, let's discuss blood. Blood is a special connective tissue. We have already read that in structural organization. Now, blood consists of two components. What are the two components here? The liquid part, which is known as plasma, which constitutes around 55 to 60% of the total blood. And then we have these formed elements. So we have plasma, the liquid part, and second, the formed elements, the cellular part. So we know there are three types of cells present here, But first, let's discuss more about plasma. So plasma constitutes 55% of the blood. It is straw colored. It is yellowish colored because of the presence of bilirubin. So what gives it straw color? It is bilirubin which gives it that yellowish color. Are these points clear to everyone? Please let me know. Then, what are the components of plasma? So 90 to 92% is water. So majority is formed by water. Then second largest component is proteins. Second largest is proteins. Now, which type of proteins are present in plasma? So first one is fibrinogen, which helps in clotting. Second is globulins, which help in providing immunity. Antibodies are also globulins. I hope this is clear. And then we have the smallest of them all, which is albumins, and they help in maintaining the osmotic balance. Osmotic balance means determining that how much blood will be present in the how much water will be present in the blood. Is that clear? Then another component, apart from water and proteins, the third component is electrolytes. So we have so many ions, electrolytes present in our blood. Then and fourth one is the nutrients. So everything that you eat first gets into the plasma, and then plasma is going to transport it to rest of the tissues. So nutrients, gases, hormones, waste, all these things are present in plasma. I hope this point is clear to everyone. All these points clear? I think it's the easiest thing we have discussed so far. Now, talking about the formed elements, we have three types of formed elements, three types of cells: RBCs, WBCs, and platelets. Is that clear? Now, RBCs are maximum. Second largest is the platelets, and WBCs are minimum in their amount. Are these points clear? Yes, please. Are these points clear? So, guys, if you are liking the revision, please don't forget to hit the bell icon and share it with your friends. Okay. And also, before I move ahead, I want to inform everyone that from Sunday onwards, guys, from Sunday onwards, we are starting with the All India mock test series from Sunday, that is 26th of April to Friday. We'll give you just one day for your relaxation so that you get NEET ready, you prepare your dress well, right? You get your things ready for your exam. Otherwise, from Sunday to Friday, you have to sit in the All India mock test series from 2 to 5 p.m. Everybody has to do that. Everybody has to do that. Are these points clear? All right. Perfect. And do let me know in every class that what is the score. You can leave your score in the comment section. I hope some of you have given this mock test on last Sunday. Those who have missed it, please make sure now it's not the time. You can't afford missing these mock tests now. Cool. Now coming on to RBCs, also known as erythrocytes, and how much are they? All right. How much RBCs are present? Between 4 to 5.5 million RBCs per millimeter cube. So these are the most abundant. There is no nucleus in RBC. So hence it is enucleated. This is the term used. It is enucleated. Why? It has no mitochondria, also no Golgi body to increase the surface area. And it contains, so it is biconcave in shape, or we call it discoidal. It contains hemoglobin, which is the respiratory pigment, and it is this hemoglobin which is actually going to help in gaseous exchange, and we have just read about it in the previous chapter, gaseous exchange. I hope these points are clear. And then the lifespan of a mature RBC is 120 days, after which, and these are formed in red bone marrow, and after 120 days it goes into the graveyard. Can anyone tell me which structure is known as the graveyard of RBCs? Yes, guys. Which structure is known as the graveyard? Supriya, I'll try to send those NCERT highlights. I'll do that. Yes, guys. Let me know. Quickly let me know. Which structure is known as spleen? Very good. Spleen is known as the graveyard of RBCs. Now see, this is how NCERT has shown RBC. Now these are the platelets, and rest all these structures are different types of WBCs. You must be able to identify these structures. Is that clear? You must be able to identify these structures. Now moving on to the least cells, the cells which are present in least amount in your blood, which is WBC, and how much are they? They are only 6,000 to 8,000 per millimeter cube. They are colorless. That is why we call them white blood cells. They are nucleated. And in the previous diagram, guys, we have seen the different types of nuclei. See, eosinophils have bilobed nucleus, while basophils have S-shaped nucleus. Neutrophils have multi-lobed nucleus. Monocytes has kidney-shaped nucleus. These are the different types of nucleus I'm telling you. And T lymphocytes have a large circular nucleus. Be it T or B lymphocyte, both have circular, and maximum space is occupied by nucleus. Are these points clear to all? So there are nucleus present, and nuclei are of different shapes. Now the main function of our WBCs is of course to provide us defense against the diseases, because of after all they are the soldiers and scavengers of the body. They are the soldiers. Now overall WBCs can be differentiated into two types, granulocytes and agranulocytes. Is that clear? So what are, as the name says, granulocytes will have granules, and agranulocytes will not have granules, no granules here, right? Now the three types of granulocytes are neutrophils, eosinophils, and basophils, based on the fact that what type of granules are present. So in neutrophils, both acidic basic granules are present, both acidic and basic. Eosinophils, also known as acidophils, because they have acidic granules, and then basophils, they'll have basic granules. What are the functions? Neutrophils are phagocytic in nature, so they will engulf the organism, the pathogen. Are these points clear to all? Okay. Then basophils will release the histamine, so they have important role to play during inflammations. They will cause inflammation. Then agranulocyte are of two types, lymphocytes and monocytes. Monocytes are again phagocytic, while lymphocytes are of two types, B lymphocyte and T lymphocyte, and they provide you they are important for acquired immunity. They're important for specific or acquired immunity. Now the third type of cells present in blood are called platelets. They are not real cells, they are cell fragments. They are deformed from the mother cell known as megakaryocyte. And how much are they? Only 1.3 to 3 lakh per millimeter cube. The lifespan is around 7 days, and the main role of platelets in our body is to help in clotting. Are these points clear? Then moving on to the blood groups. So guys, your RBCs, the surface of RBC has certain proteins, glycoproteins present on them. Now these glycoproteins are known as antigens. Now based on the type of antigen present on your RBC, we have different types of blood groups. See, your antigen and your blood group are exactly the same. They are exactly the same. Whichever antigen, that blood group will be present. Now O blood group basically means 0. There is no antigen in case of blood group O. Clear? Now antigens are already present, and antibodies are present in the plasma. Antigens are on the RBC surface, antibodies are in plasma. Now whatever antigen you have, antibodies are present reverse to that. For example, if I have antigen A, then my plasma will have anti-B antibody. Similarly, in case of antigen B, my plasma will have anti-A. If the person has both the antigens, there will be no antibody, while if the person has no antigen, there will be both the antibodies present. So accordingly, we can decide who is the donor group and who is the recipient group. As a matter of fact, let's jump on to the conclusion. O is known as the universal donor. Why? Because of no antigen. While and AB is the universal recipient because of no antibody. I hope these points are clear to you. Now guys, apart from ABO, we have another type of antigen also, which is called RH. If the person has RH, the person is RH positive. If the person doesn't have RH group, RH antigen, that person is RH negative. Now this RH is actually important, number one, of course, during blood transfusion. So you can give negative blood to the positive person, but you cannot give positive blood to the negative person. Is that clear? And secondly, it is during the pregnancy. So in case, guys, see, in case, let's talk about the RH. Okay, now see here, in case, yes please, in case the mother is RH positive and the father is RH positive, baby will be positive. Both negative, baby will be negative. Now if mother is positive, father is negative, even one is positive, baby will be positive. Is that clear? Now mother negative, father positive, baby will be positive. Now the point is, just see here, the blood group, sorry, the RH antigen of mother and baby should rather match, right? Because the baby is going to develop in the mother's body. Now see here, both have positive positive, both have negative negative, both have positive positive. The point is when, and the problem is when mother has RH negative blood group, father had RH positive blood group, and that led to baby having the positive blood group. Right now, this is the point. This is the situation, right, where RH incompatibility arises because mother is RH negative, baby is RH positive. This is RH incompatibility. But in this case, what happens? First pregnancy would be normal. First pregnancy would be normal, but all the subsequent, all the future pregnancies, starting from second onwards, all the future pregnancies will lead to stillbirth. That is, dead baby will be born. That is because of erythroblastosis fetalis. That is because of erythroblastosis fetalis. So the baby will suffer from severe anemia, and severe jaundice. This will lead to the death of the baby. Are these points clear to all? Please let me know. Please let me know. Then guys, we have lymph or the tissue fluid. So the tissue is formation. Tissue is formed from blood and then poured back into the blood. So it is basically plasma that filters out through capillaries along with some WBCs. It is plasma along with some WBCs that comes out through capillaries, right? It has less proteins than plasma and contains lymphocytes, which WBCs majorly lymphocytes. Now moving on to the main structure, which represents our circulatory system, that is human heart. Now where is heart present? It is again present in the thoracic cavity, right between the lungs, but slightly tilted towards the left side. Now just like lungs had a protective covering known as pleura, double membrane, our heart also has a covering known as pericardium. This is also double membrane. Outer one will be known as parietal and the inner one will be known as visceral, and in between the two there will be pericardial fluid. Which will prevent friction and keep it hydrated. Are these points clear to everyone? Please let me know. Now see, this is quite easy to, I think this is something we already know. There are 4 chambers. How many chambers are there? There are 4 chambers. So this side represents the right side. This is the left side. So see the structure of heart. So this is vena cava, which is bringing the deoxygenated blood, right? So this brings the deoxygenated blood from the whole body. So right side contains the deoxygenated blood. And then aorta. Aorta is going to take away the oxygenated blood to the rest of the body, right? Then we have this right auricle, and in right auricle, guys, we have the sino-auricular node. So this is where the impulse is generated. We call it the pacemaker. This is the pacemaker, SAN. Where is pacemaker present? In the upper right corner of right auricle. Yes, so this is present in the upper right corner, while towards the lower left corner is present AVN. The AVN. Now both the structures are present in right auricle itself. So see the location. Clear? Now this being your right ventricle, this is your left auricle and left ventricle. Now do you see these thread-like structures here? These thread-like structures are known as chordae tendineae. So basically they hold on to the valves and prevent them from opening in the opposite side. So this is your tricuspid valve. These are your bicuspid valves. So right auricle, right ventricle connected through tricuspid valve, and left auricle, left ventricle through bicuspid valve. Are these clear? And from the lower side, in the ventricles are present thread-like structures to hold on to the these valves to prevent them from opening in the opposite direction. These threads, collagenous threads basically, they are known as chordae tendineae, and they're actually very strong fibers. Now, other than these guys, see, these chordae tendineae are present on these structures. They are present on these structures, which are known as columnae carneae. The structures through which chordae tendineae are present, they are known as columnae carneae. Is that clear? I hope these points are clear to everyone. And here we also see all in yellow, everything drawn in yellow. See, starting with sino-auricular node. This is sino-auricular node. Let me keep the color yellow. So this is sino-auricular node where impulse generates. This is AVN, which is going to amplify it and transfer the impulse further. And from here arises the auricular ventricular node, which then breaks into the right and the left fiber. Is that clear? And branches, and then into the Purkinje fibers. And towards the apex, it breaks into the Purkinje fibers. So these are the fibers. And these fibers which help in transferring the impulse, they are actually modified muscle fibers. These are all modified muscle fibers. Which muscle? Modified cardiac muscle fibers. This was a PYQ. This was a PYQ. Are these points clear to all? Now, what are the functions? They transport an interstitial fluid to immunity. This is something else. Leave this, guys. Now, coming on to circulatory pathway. So, we know there can be two types of pathways. We have already read the animal kingdom. If capillaries are present, then that is known as closed circulatory system. The blood flows only in the blood vessels. So arteries are connected to veins through capillaries. So blood will only be flowing through these channels. This is known as closed circulatory system. And we see closed circulatory system in only two phyla, Annelida and Chordata, while rest of them either don't have a circulatory system, and if it is present, it is of open type. So blood flows through the body cavity. So wherever there is space, blood will be flowing into it. Humans have a closed blood circulatory system, and we have a double circulation, and that too complete. Double circulatory system, I hope everybody knows, and because there is no mixing, we call it complete double circulation. Are these points clear to all? Are these points clear to everyone? Please let me know. Alright, moving on to now the blood vessels. So we have in all three types of blood vessels, guys. Arteries, which usually carry blood, which usually carry oxygenated blood, but there is an exception, except pulmonary artery, right? The standard definition is it is a blood vessel which carries blood away from heart, right? While veins carry blood towards the heart, usually carries deoxygenated, but exception is again except pulmonary veins. Pulmonary veins. Are these points clear? And while capillaries are the smallest blood vessels, thinnest blood vessels, where actually exchange of materials would take place. Now, see the structure. Arteries are thick and elastic. See, because they are carrying blood away from the heart, so blood is flowing under high pressure. Blood is flowing under high pressure. So that is why it high blood pressure. That is why they need thick walls and elastic walls. Is that clear? But in case of veins, since the blood pressure is very low, therefore they need valves to prevent the backflow, and the walls are also thin. Is that clear? And the capillary is only made of single wall, single layer, and that single layer of cells is known as endothelium, and it represents the squamous epithelium. Why squamous? Because exchange has to take place, diffusion has to take place. Are these points clear to all? I'll be coming to the T wave. I'll be coming to the ECG very soon. Coming to ECG. Yes, guys. Are these points clear? So see, outer layer of any blood vessel, that is vein or artery, is known as tunica externa. It is made up of white fibers. It is made up of white connective tissue. Then the middle layer is tunica media, and it is made up of smooth muscles. This represents a smooth muscle layer, right? And then we have tunica interna. This tunica interna is made up of the squamous epithelium known as endothelium. Now, basically, you see here, The major difference in the thickness of artery and the vein is because of tunica media. In case of artery, tunica media is broad. Can you see the red portion? It is very broad. It is very thick. While in case of vein, this is very thin. So the basic difference in the wall is because of tunica media, while tunica externa and externa and interna are just the same. Are these points clear to all? Please let me know. Now moving on to the very important topic, guys, which happens to be your cardiac cycle. This is your cardiac cycle. So cardiac cycle, the duration is zero point 0.8 second. So between two heartbeats, between all the events that occur between two heartbeats are known as cardiac cycle. Okay, so what happens really? So it begins with joint diastole, then there is ventricular systole, and there is atrial systole. So starting here with the atrial and ventricular diastole. Now this is known as a joint diastole. When both atria and ventricles are in the relaxed state, we call it joint diastole. Now when all the vessels, all the chambers, atria as well as ventricles, they are relaxed, so there is negative pressure. So what is negative pressure within the heart? Now the blood from the blood from your vena cava and your pulmonary veins, they are entering into the heart. Now since there is negative pressure, so AV valves are open. AV valves are open, but SLV valves are closed. So what are AV? That is tricuspid and bicuspid valves. These are open. Negative pressure in heart, and this leads to filling of the ventricles. So 70% of the filling of ventricles is going to occur during joint diastole. Is that clear? 70% of ventricular filling would occur here. Are these points clear to all? Please let me know. You're joining the session now. Why, Ansh? Never mind. Just go with the flow now, and then you can revise rest of the just one chapter done so far. We are going discussing the second chapter. So you can discuss these two chapters because this is almost on the verge of getting completed. You can do that later. All right, guys. So now see, in the joint diastole, negative pressure in heart, TVBV open, and SLV close. And there is negative pressure, so blood from the vena cava and pulmonary veins is getting into the atria, and since TVBV are open, it is going into the ventricles. So 70% ventricular filling is going to take place during joint diastole, and this is slow in speed. Now once 0.8 seconds, 0.4 seconds are done. Joint diastole is for 0.4 seconds. 0.4 seconds are over. Now, SAN will produce an impulse. So, when SAN produces an impulse, see here, SAN produces impulse here at the end of 0.4 seconds, what will happen? Atria are going to close. So, both the atria will close. This is known as atrial systole. Now, while the atria will contract, your ventricles are still relaxed. Is that clear? So this atrial contraction will go for only 0.1 seconds. Now what will happen? The blood which is entering the atria will forcefully be pushed towards the ventricles. So in atrial systole, 30% ventricular, remaining ventricles will be filled. 30% ventricular filling. 30% ventricular filling would take place during this phase. Are these points clear? Yes or no? 30% will fill during this phase, and now because the atria contracting, it is putting pressure on the atria, so this filling is fast. Are these points clear? Please let me know. Everyone, this filling is going to be fast. Now it just stays for 0.1 second, and quickly the wave of depolarization is transferred to the ventricles. Right now what happens during this? Ventricles are going to undergo contraction. So ventricular systole. Now while ventricle will contract, atria will go in a state of relaxation. Please let me know that these points are clear to you. Now ventricular systole is going to show very important events here. They are going to take very important events during the ventricular systole. So first of all, the first thing that happens during ventricular systole is that semilunar valves will open. So is that clear? Sorry, no. I have to write it down. Okay. So first thing that takes place is that TV and BV will close. TV and BV, that is tricuspid and bicuspid valves, are going to close. This is the first thing that would happen. Now when tricuspid and bicuspid will close, first heart sound will be produced, that is lub. Is that clear? Then after TBPB closes, pressure rises, pressure increases within the heart, and this would lead to opening of semilunar valves. Opening of semilunar valves. This is the second thing that would happen. When semilunar will open, blood will go out. So from right auricle, blood will go into the pulmonary artery to the lungs, and from the left ventricle, the semilunar valves will open to push blood through aorta to the rest of the body. Is this clear? So blood will go out, blood ejection occurs. Please tell me these points are clear to you all. Is this point clear? Is this point clear? Then the third thing, after the blood has gone out, see, ventricles are not going to get empty here. But when maximum blood goes out and there's a fall in the pressure, again, what will happen? Now AV, BV valves will close. Okay, now this is already written. Sorry, this is written here. What has to be the first thing? Now, what will happen when pressure will increase, blood will go out, and now the pressure is going to fall again, right? After the blood has gone out, pressure is going to fall again, which will lead to closing of, the third thing is closure of SLV. Closure of SLV. Now closure of SLV will produce the second sound, which is dub. Closure of SLV will produce a sound which is called dub. Is that clear? What is isovolumetric contraction? See, this is isovolumetric contraction in the initial phase when TBBV closes, right? Isovolumetric contraction. This is in the beginning of the ventricular systole. Isovolumetric contraction will lead to TV and BV closure. Is it clear? Then second phase of blood ejection. And then towards the end of the ventricular systole, isovolumetric relaxation. Isovolumetric relaxation. Are these points clear? And there will be closure of SLV leading to the second sound, that is dub. Is this point clear? Yes, please. Please let me know that these points are clear to all. So see, clearly written which sound will be produced. Now these are the two normal sounds. Also remember, both the sounds are produced during ventricular systole. Both the sounds are produced during ventricular systole. Is that clear? Lub during the beginning of ventricular systole and dub during the end of the ventricular systole. Please let me know that these points are clear to you. Now question is, which sound is loud and which is softer? So guys, okay, I'll write it down here. So lub and dub. See, lub is the first sound. This is the second sound. This is due to the closure of AV valves, right? That is TV and BV. This is due to the closure of SLV valves. Now, talking about lub, this is during the beginning of ventricular systole. Told you, beginning of ventricular systole. While this is due to towards the end of ventricular systole. Now, the question that has been asked in the chat, which one is louder? So this one is louder, but low pitched, and it is of longer duration. This is of longer duration. These are the points: long, loud, low pitched, that is shriller, right? And this one is opposite. It is short, it is sharp, and it is high pitched. Are these points clear to all? Please let me know that these points are clear to everyone. Thank you, universe. Okay, are these points clear to all? Moving ahead now to ECG, very, very important. First of all, ECG stands for electrocardiogram. not graph, not graphy. It is electrocardiogram. Just get it right. In case somebody is using the old NCERT, please get it right. G stands for electrocardiogram. Now this is that reading that comes to us. So what is represented here is electrocardiogram. Is that clear? This is electrocardiogram. Now the device used is electrocardiograph. The machine that we used is named as graph. Is that clear? And the process is, mechanism is, or the study is known as graphy. It is graphy. Now, ECG, please see, represents the electrical activity of heart. It represents the electrical activity of heart. Clear? Now we see some waves here. The P wave represents the atrial depolarization. See, it is not the same as atrial systole. Systole will occur right after depolarization, right? So this represents the electrical activity, while systole is the mechanical activity. So remember that there's a difference between the two. So P wave represents the atrial depolarization, while QRS, we call it QRS complex, it represents the ventricular depolarization. And NCERT has clearly said that actual ventricular systole will start shortly after the Q wave, right? Shortly after that. Are these points clear to all? Please let me know. Please let me know. And guys, I have completed NCERT reading. I've already completed NCERT reading in 4.5 hours. Please make sure that you still revise NCERT before going. You can increase the speed of the video, and you can revise NCERT in around 2.5 to 3 hours. Clear? I have highlighted the PYQs. Those of you who have missed that session, please make sure that you do that before going for the real NEET exam. And from Sunday onwards, AIMT, All India Mock Test Series, is starting from 2 to 5 every day. You have to take that paper before going for the NEET exam. Is that clear? All right. Moving on to the next. Yes. So QRS complex represents the ventricular depolarization, and T shows the ventricular repolarization. That means after which ventricles will go back into the state of relaxation. Please tell me that these points are clear. Now the duration, now the length of P, Q, R, S, all these are of clinical significance, and they help us in diagnosing the disease. They help us in diagnosing the disease. Is that clear? You got only 328 for biology in today's mock. King, you let me know in the chat, in the comment section in detail what was the issue, what are the problems that you're facing. I'll go through it and I'll answer your question. Is that clear? All right. Now, guys, if you have to count the number of heartbeats, see here, I'll give you a simple formula. So, number of heartbeats is equal to the number of QRS complex. QRS complex. So when you have to count the heartbeat from the electrocardiogram, you just encircle the QRS complexes. Also, it is equal to the number of cardiac cycles. So the cardiac cycle is also just the same. Is this point clear? So there are two other terms. Stroke volume. So what is stroke volume? Stroke volume is the volume pumped in one heartbeat. That is during one cardiac cycle. So which is equivalent to 70 ml. But how many times your heart beats per minute? That is the heart rate. So if you multiply heart rate with stroke volume, that is going to give you the cardiac output. Please let me know that these points are clear. So if I multiply 72 by 70, it will be roughly 5240, something like this. So around 5 liter. That means the whole blood is pumped through heart in 1 minute. Please tell me that these points are clear. Manisha, please write down this message in the comment. I'll answer it there. All right, guys. Then moving on to the double circulation. Double circulation, guys, is that blood enters the heart twice. So there is pulmonary circulation and there is systemic circulation. Pulmonary is between heart, lungs, and back to the heart, while systemic circulation is between heart, body, and heart. Is it clear? It's between heart and the body, and pulmonary represents heart and the lungs. So the advantage is there is no mixing and it shows efficient oxygen supply. Now, how is cardiac activity regulated? So again, your brain will come in, and but brain is not directly going to regulate the heart activity. See, heart is autorhythmic. Your heart is autorhythmic. Also, it is called myogenic. The muscle of the heart will produce the impulse on its own. So the heart activity, cardiac activity is not directly regulated by your brain or your hormones. So impulse has to be generated within the heart, that is in SAN. Now, if SAN is producing the impulse, if SAN is working normally, then your brain and your endocrine system can influence it. They can influence it. See, heart is myogenic. It has to be produced in pacemaker and relayed by AV node. But if these are working properly, then your sympathetic autonomic nervous system can increase the heart rate, while parasympathetic can decrease the heart rate. Similarly, now sympathetic works along with adrenaline and noradrenaline. So both of them are going to increase the heart rate, while acetylcholine is going to reduce it. Please let me know that all these points are clear to you all. Please let me know, guys, why your marks are not improving. So let me know in the comment section. I will be answering your doubts there, definitely. And one very clear thing is that you should keep on giving mock tests, so that, and you should analyze every mock test. It's very important. It's not just about giving. You have been giving these tests from past 2 years. The point is you must know uh, that you must do the analysis. So right after the mock test, you will get a video solution also, where you can analyze where you were going wrong. What were you thinking while doing the paper? So analysis is the key. Is that clear? Analysis is the key. You're making silly mistakes, maybe because you're getting scared of the exam. Are you? See, exam is something that everybody is scared of, but then you have to calm down your nerves, because even if you are scared, still nothing won't happen, right? You won't be able to crack if you will be so scared. So just relax. Dekhi jayegi. We'll see what happens. Just do your best and leave the rest, right? And doing your best means getting yourself best prepared, revising everything, but with a calm mind, okay? And giving all the mock tests. Analysis is the key, guys. Analysis is the key. So for that analysis, you will be provided video solutions right after the exam. That is very important. So that is why, see, I can get all the complex notes here, right? I can get all the detailed notes. But I am revising these units only by the help of these quick points, so that we can hit on the target, right? That is why I'm bringing these short notes here, so that you can just recollect the exact word in exam. So make sure that now you are just learning these things, and not going through the detailed notes. At the max, if you want to go for the reading, then the sessions are already done. You can just read it. Imagine you just have to give 2.5 to 3 hours to complete your one NCRT. Just 3 hours for completing one NCRT. Can we do that? We can do that. Sit tonight and do that. 6 hours and your whole two books will be done. Yes or no? What do you want more than this? And then after reading these NCRTs, go for your mock test. Is this clear? And the mistakes that you're making in your mock test, please make sure that you mark them in your NCRT, so that every time you revise, you know that, or write down separately. Is that clear? Alright, guys. Moving ahead to the next chapter, and that is excretion. That is excretion. What is excretion? Removal of metabolic waste. And what type of metabolic waste? Nitrogenous metabolic waste. Now, these nitrogenous metabolic waste can be they're of many types, but they're chiefly of three types: ammonia, urea, and uric acid. Ammonia is the most toxic, while uric acid is the least toxic. Right? Ammonia needs maximum water for removal. Uric acid needs least water for its removal. So since it is least toxic, it can be stored for a long time. You cannot afford storing ammonia in the body. It is highly toxic. So it has to be removed as quickly as it is produced. So cannot be stored. While you can see amongst the two extremes, that is ammonia and uric acid, we have urea, which is an intermediate. It is toxic, but not very toxic. It is intermediate when it comes to toxicity or requirement of water for its removal. It can be stored to certain extent beyond which it becomes toxic. Are these points clear? Now, very, very important, which organisms are ammonotelic, which organisms uricotelic and ureotelic. So one thing is for sure, as we know that ammonia requires large amount of water for its removal, so 100% they are always aquatic. Ammonotelics are always aquatic. But I did not say that all aquatic are ammonotelic. Remember, see, ammonotelics are always aquatic, but all aquatic organisms are not always ammonotelic. Now, what are the examples? Your many bony fishes, aquatic amphibians, they are ammonotelic. Is that clear? Then, you in ureotelic, we are ureotelic humans, adult amphibians. So while it is in tadpole stage, it is a monotelic, and later on the adult frog becomes ureotelic, and other amphibians too. Coming on to uricotelic. Now these uricotelic organisms are those which have least water. So if the organism has least water, they have evolved to become uricotelic. For example, now see birds. They can't afford carrying a lot of water in the body since they have to fly, right? Reptiles are also first land animals, and then they live in dry areas also. They are also uricotelic. Insects also don't have a lot of water. They are also uricotelic. Are these points clear to all? I have already made a video on cockroach, guys. Tonight I'll make it on frog. Quickly, you can revise that cockroach now. Also for plant families, I've made a video on plant families where you can revise the plant families by just remembering 5 different floral formulae. Just 5 floral formulae and you will revise it all. Is that clear? So make sure that you watch those videos. Now coming on to here, the human excretory system which consists of a pair of kidneys, then a pair of ureters, one urinary bladder, and then urethra. Is it clear? Starting with kidneys, bean shaped, located between T12 and L3, that is 12th thoracic vertebra and 3rd lumbar vertebra. Are these points clear? And you know that the right kidney is slightly lower. Right kidney is slightly lower than the left one because right above this lies the liver. So liver is going to push it slightly downwards. Are these points clear to everyone? Please let me know. Now, like any organ, your kidney can be differentiated into two parts. The outer part will be known as cortex, while the inner part will be known as medulla. Is that clear? Now the medulla, see, this is cortex. So this is cortex and medulla. Now the medulla part is differentiated into these structures these structures are known as pyramids. These are known as pyramids. These pyramids will open into these funnel shaped structures called papillae, and then papillae will open into small and then large ducts. These ducts are known as calyces. These are the ducts, and then finally all the ducts will together open in this broad region which is known as pelvis, renal pelvis, and from pelvis arises your ureter. Are these points clear to all? Right? Now the unit of kidneys is known as nephron, the structural as well as the functional part. There are two components, there are two parts of nephron, right? One is glomerulus and another is the renal renal tubule. Is the renal tubule. Is that clear? Now glomerulus is a group of capillaries, blood capillaries, while renal tubule consists of Bowman's capsule, then PCT, Bowman's capsule, PCT, loop of Henle, DCT. All these constitute the renal tubule. Now see, collecting duct is not a part of a nephron. It is a tube in which many nephrons are going to open. Is this clear? Now capillaries, that is and Bowman's capsule. Wait, glomerulus and the outer one is the Bowman's capsule. Together is known as Malpighian body or renal corpuscle, and they all have different functions to play. Are these clear? Now, if we see, there are two types of nephrons, guys. There are two types of nephrons. One is the cortical nephron, and the other one is the juxtamedullary nephron. So, what do we understand by this? See, this is the cortex region, this is the medulla region. So now we have two types of these. See, this is the Bowman's capsule, this is the PCT, right? And this is a short loop of Henle, and then this is the DCT. Is this clear? Now, this one is known as the cortical nephron, and 80% of your nephrons are cortical. While we have another type of nephron in which, see, Bowman's capsule will always be present here, PCT will be present in the cortex, DCT will be present in the cortex, but loop of Henle goes deep into the medulla. Now, such nephron is known as juxtamedullary, and these are only 15 to 20% of the total. Is this clear? Long loop of Henle. Now, long loop of Henle means they will be involved in water conservation. Water conservation. So, long loop of Henle means they also will have a long vasa recta. They will also have a vasa recta. While vasa recta is absent here, and no water conservation. No water conservation. Please tell me that these points are clear to you all. Please tell me these points are clear to you all. And then, guys, we have a special structure in the kidneys, that is juxtaglomerular apparatus, the function of which we are going to study in a while. Now, see, talking about the function of kidney. Kidney is known for urine formation. So, basically, that function is done by your nephrons, and we have 1 million nephrons in each kidney. Now, urine formation occurs in 3 steps. It occurs in 3 steps. What are the 3 steps? First, filtration. Second, reabsorption. Third is tubular secretion. So, glomerular filtration, also known as ultrafiltration. Why? Because it is going to occur under high pressure. Blood is filtered under high pressure. So, when blood is filtered under high pressure, everything comes out. See, this is your Bowman's capsule. When there is a high pressure, everything is going to come out. Everything is going to come out except your cells, blood cells will not come out. Proteins will remain behind. Rest, all your nutrients, all your hormones, all the vitamins, minerals, like your nutrients itself, all the salts, all water, everything is going to come out. Complete glucose, all the amino acids, everything is going to come out. Is this clear? Except for these 2 things, which are cells, blood cells, and proteins, they will not come out. Now, your kidney receives around 1/5 of the total blood of the body, which is around 1100 ml. 1100 ml, 1/5 of the total blood. You have around 5000 to 6000 liter of blood. So 1100 ml of blood passes into the kidney every minute. Is it clear? Now, out of that blood, 125 ml of filtrate is produced. How much filtrate is produced every minute? 125. Now, this is known as glomerular filtration rate. So, this is the amount of filtrate produced by your glomerulus or your Bowman's capsule in 1 minute. So, overall in a day, it would be 180 liter. Is it clear? But basically, what happens during glomerular filtration is everything, useful as well as non-useful things, substances, non-useful substances, both come out. So as far as non-useful has come out, it is a good thing. But we see that all the nutrients have also come out. 100% water came out. All the glucose has come out. Are you getting? All your salts have come out. So there is a need of a process which can return the useful substances back to the blood. So here comes the tubular reabsorption. Tubular reabsorption will occur in PCT, loop of Henle, DCT, and the collecting tubules. While glomerular filtration occurs only in the glomerulus and Bowman's capsule. Bowman's capsule. I hope these points are clear. Most of it occurs in the PCT. So that is why PCT is specially designed for maximum reabsorption in the form of having brush bordered cuboidal epithelium. Is this clear? So maximum reabsorption is going to take place in PCT. That means maximum water or sodium, all are going to be absorbed to the maximum extent here in the PCT itself. 100% water, all these are reabsorbed. 100% glucose reabsorption and amino acids are reabsorbed here. Please let me know that these points are clear to all. Is this clear? Then there is another process which is called tubular secretion, where we put back the waste material back into the nephron. Now see, this diagram will explain everything. Now see, it clearly represents that structures which are present in the cortex area. See, cortex has Bowman's capsule, it has PCT, it has a slight part of loop of Henle and the DCT, right? While collecting duct is present in both cortex and medulla. Is it clear? Now see here, the diagram from your NCERT clearly shows. Now see, these substances, H positive ammonia, K positive, H positive, the arrowhead towards the nephron means you're putting it back into the nephron. This is called tubular secretion. This is called tubular secretion. While the arrowhead away from the nephron, all these things, they represent the tubular reabsorption. Now, through this diagram, you can clearly see and remember which components will be reabsorbed from PCT and DCT, and which ones will be which will be secreted in the nephron from PCT and DCT. Are these points clear to all? Are these points clear to all? While we see here that the descending loop of Henle is permeable only to water. So water is coming out. It is permeable only to water, right? While the ascending limb is permeable only to sodium. Are you getting these points clear? Now see, concentration of urine is important, and the mechanism involved in concentration of urine is called counter current mechanism. Here two main structures are involved. One is loop of Henle and second is vasa recta. Second is vasa recta. The two structures involved are loop of Henle and vasa recta. Is it clear? Now, as we go deeper into the cortex, the osmolarity goes in increasing. So ultimate thing that you have to remember, see two things. Descending limb of loop of Henle is permeable to water, while the ascending limb of loop of Henle is impermeable to water. And the medullary osmotic gradient is maintained by NaCl and urea. These are the two things which are maintaining the osmolarity. Now moving on to the function of kidney. Other than, see, how is kidney function regulated? How is kidney function regulated? Through these hormones. So first hormone is ADH. It is going to help in water reabsorption. How? We'll see in a while. Second is aldosterone. It is going to help in sodium reabsorption and increasing the blood pressure, right? While ANF, atrial natriuretic factor, okay, this is going to decrease the sodium reabsorption or decrease the blood pressure. This is secreted by atrial walls of heart. Atrial walls of heart. Now, how does every hormone function? So you know that your posterior, so hypothalamus produces it, and posterior is going to release it, released by posterior pituitary. Posterior pituitary is going to release it. Now it acts on the DCT. ADH acts on DCT and collecting tubules and help in water reabsorption. Okay, it will help in water reabsorption. Now when is this happening? This happens during summers or when there is excessive water removal because of vomiting or any reason of dehydration. This will stimulate the ADH production. Is this clear? Then if your blood pressure is high, then your heart walls are going to secrete ANF, and ANF is going to decrease the sodium reabsorption, thereby lowering the blood pressure. While if we talk about increasing the blood pressure, we have this detailed RAS mechanism. We call it RAS mechanism. So renin, angiotensin and aldosterone system. So we have renin, we have angiotensin and we have aldosterone system, hence the name RAS. So what really happens when there is drop in the blood pressure? See here, when there is a drop in the blood pressure or the GFR decreases or blood pressure decreases, then your kidney is stimulated to produce renin. Your kidney releases renin. This renin enters the blood. It enters the blood, renin in blood. Your blood already has angiotensin, which is produced by liver. Liver produces angiotensin. Is this clear? Now, angiotensin in blood will be converted into angiotensin by the presence of renin. By the presence of renin. Angiotensinogen is the inactive form. It will be activated by the presence of renin. Right? Now, more than angiotensin 1, angiotensin 2 is more potent. So, when angiotensin 1 is formed, it is converted into angiotensin 2 by ACE, which is secreted by the lung capillaries. Clear? Now, once angiotensin 2 is formed, it is naturally a vasoconstrictor. It is a strong vasoconstrictor. When the vessels are constricted, see, it's written here. Why do we have to write it again? It is a vasoconstrictor. It acts directly on the blood vessels, constricting them. When the vessels are constricted, blood pressure increases. So that's the aim. Is that clear? Then aldosterone is going to increase the water reabsorption and sodium reabsorption. And this is further going to increase the blood pressure. So RAS mechanism helps in increasing the blood pressure. It increases the blood pressure. Please tell me that these points are clear to you all. And last not the least, guys, we have micturition. Urine is stored in the urinary bladder, controlled by the central nervous system. So there is voluntary control as well as an involuntary control. And we call that whole set of reflex as a micturition reflex. So message from the stretch receptors. So message from the stretch receptors of urinary bladder. It reaches your central nervous system. And then central nervous system again sends message to the walls of bladder so that they can contract, and it sends signal to the sphincters, urethral sphincters, so that they can relax, and that is how micturition takes place. Please let me know that these points are clear. Other than, guys, your liver, your kidneys, lungs, liver and skin, they are also excretory in nature and they secrete different substances. Now, last not the least, let's discuss the disorders. We have uremia, in which there is over accumulation of urea. I told you some urea is stored in the blood, but more than that, beyond 34 or 37 milligram, it becomes toxic, and that condition is known as uremia. It will happen when there is a kidney failure. It occurs due to kidney failure, right? Then kidney stones. So deposition of calcium oxalate deposits in kidney leads to kidney stones, or we also call them renal calculi. And then glomerulonephritis, itis, means inflammation of glomerulus. So these are the disorders. So guys, we'll have to stop here. I have been informed by the team to end the session. I think there is some thing coming up. So I think we'll have to complete the human physiology in part 2. So I'll join you again. So make sure, guys, by then these 3 chapters are done, and you don't forget to take your mock tests from Sunday onwards every day 2 to 5 p.m. Okay, I'll see you in the next session of human physiology. I think there is something coming up. So I'll have to end the session now. Thank you so much for joining me, and wait for the second part of human physiology.